The Horn of Africa is the easternmost peninsula of the African continent, bordering the Red Sea to the north and the Somali Sea to the east, as well as the Indian Ocean to the south. Politically, the Greater Horn of Africa comprises of 11 countries, each with diverse communities. The region is home to a large variety of wildlife, including the Big Five, and it is also known as the cradle of mankind, given that the earliest hominin remains have been found here. Culturally, the region is rich and diverse, with livestock keeping being a common factor across all the communities and culture. The bond between livestock and man is deep and intimate, stretching back through history. The region holds about 10% of the world's herd, with an average contribution per country of approximately 10% of the GDP. Livestock also contributes indirectly to agriculture in terms of drought power and manure provision, as well as being a key pillar of socioeconomic significance to the communities involved. In particular, cattle are a source of nutrition, draft power, or a stock of wealth. While camels can be employed as a mode of transport as well as a source of nutrition. Last but not least, small livestock such as goats, pigs, and sheep can be utilized as a stock of wealth, providing much needed financial empowerment for the women and girls in the region. However, livestock farming is threatened by viral diseases such as foot and mouth disease. To tackle this disease, 4 researchers from Belgium, Kenya, and Ethiopia came together formulating a joint proposal which was submitted to VLIR, and this proposal was namely initiating a training network and capacity building to improve control of foot and mouth disease in Kenya and Ethiopia.
Good morning, good day, and good afternoon, and good evening. Welcome on our VLIR US Community Talk. And the Community Talk will focus on our Teams project, abbreviated KEAT. It's between Kenya and Ethiopia, and it's on diagnostic and training networks for foot-and-mouth disease virus. I'm Jan Paeshuyse. I'm Professor of Pathogen Interactions in livestock at the KULeuven, and the KULeuven is situated in Leuven, 30 kilometers from the capital of Belgium, that's Brussels, in the center of Europe. And in Leuven we have a long history of a university that's already there for since 1400s. We are also renowned or known for our brewery, Stella Artois. And myself, I'm affiliated with the Faculty of Bioscience Engineering. And the Faculty of Bioscience Engineering is about 2,500 students that follow 14 master programs and has 3 research departments. I'm affiliated with the Department of Biosystems, and within the department I'm associated with the Division of Animal and Human Health Engineering. Within that division my lab is situated, and what we do is we focus on interaction of infections with farm animals. We try to learn something about these interactions and then translate it to medication. Now, to give a bit of background, so I started my PhD in 2000, and I actually worked on the discovery of antivirals for the treatment of hepatitis C virus. So that was in the Faculty of Medicine of the KULeuven, and at the renowned Rega Institute, that is actually the cradle of tenofovir, where Eric de Klerk, among others, discovered tenofovir as an HIV inhibitor, and it's currently still a leading anti-HIV drug worldwide. But for me, it was the aim to continue that legacy, and I focused on hepatitis C. Now that's that's from the Flaviviridae, and at the time in 2000 there was no cell culture model, so we looked for an alternative, a surrogate, and we found that in the pestiviruses. It's from the same family of viruses, but they target hepatitis, and they target livestock, cattle, pigs. An example here, infection classical swine fever, and how these outbreaks are contained. And we discovered this molecule called PPIP, small molecule, that was synthesized by an Austrian chemist called Gerhard Pustinger, and that molecule was active against PVD, and later on it proved also active against hepatitis C. And this is actually kind of the key or the fundament that is used to build the laboratory of host pathogen interactions. Now, there are two main lessons that I want to share that I learned during my PhD and during my postdocs. And one, it came from Alan Rudolph, who said that we can no longer afford to develop products specific for a specific agent. Too many threats, and these threats are too dynamic. And the other one from Michael Kahn, a couple of years later, 2014, I think. So we should learn from nature to set as a goal to achieve full control of viral infections instead of pursuing a total cure. And this is actually again reminiscent with the COVID outbreak. Is in a record tempo we developed specific agents, vaccines, and still developing antivirals that were geared to treat or to prevent the SARS-CoV-2 infections. But knowing that if you take this flying fox, for example, that it carries many more viruses, and that because it's able to fly, its immune system is configured differently, allows viruses to replicate and change without the animal becoming sick. How are we going to cope when the next virus comes, and the next virus, and the next one? And that's a main thing that that was a question to found the laboratory of host pathogen interactions. So we study these interactions. Some will say we study immunology. We do that from the bench to the farm set, so different levels of complexity for different pathogens, and today we focus on viruses and for different livestock species. What we learn from these interactions, we of course hope to translate into vaccines, immune modulators, or antibodies, serotherapy, and one of these workhorses in our lab is phage display. Now, there's a big variation if you compare different hosts on how they respond on pathogens. For instance, for horses, if we compare to the donkeys and zebras, they all respond differently to African horse sickness, also to malignant catarrhal fever, gnus and cattle, and for today, foot-and-mouth disease. There's also a difference between how buffaloes, for instance, respond to foot-and-mouth disease and our domestic cattle. For many of you, these are exotic breeds. For me, that is an endogenous breed. And these differences are there, and they actually hamper productive breeds for being or reaching their maximum capacity. If you then look at individuals, you also see a difference in how these individuals respond. And there's age, gender that account for such variations, also genetic and epigenetics, but stress, nutrition, environmental conditions also impact this response. And yet we develop a single solution for all these animals. Not only a single solution for the pathogen, but also a single solution for different individuals. And that's something to think of. How can we kind of tailor these solutions to match better the host's immune system or its immune response. And that's why we focused on the Horn of Africa. Lots of animals, lots of livestock there. Probably the biggest livestock concentration in the whole of Africa. So lots of individuals, lots of breeds, a lot of genetic variation. And for the mouth disease, a peculiar situation there. You have a number of topotypes and serotypes circulating there. You have the dynamics with wild animals, with wildlife, and that makes it an ideal place to study and to try to find out these differences and to translate them in usable approaches. There was a second reason why we initiated the team project, and that has to do with biological reagents. It's about being able to have access to these reagents. And this is just a scheme from Stephen Odongo from Uganda, and that also was a student IPMB program. And you had this wheel of progress. But usually where you get stuck is when you want to turn theory into practice, you need reagents. And there you have stuck. So why not use an existing method? If you go back here in Europe, and for me it actually goes back to my childhood when I want to have a lab in a backyard shed. And to get these reagents, you had to look up how you could make them yourself because they were too expensive. And once upon a time in Europe or in Belgium, there was a time that there was no life science supporting industry, which meant that you had to make your own reagents in the lab. Restriction enzymes, polymerases had to be produced in the lab because they weren't available off the shelf. And it is that knowledge, although it's a different grade and a different way of convenience, that might be very useful to mitigate this issue that many of you are encountered with. Empty shelves, empty refrigerators, empty freezers, you all know that. As long as it's empty, there is no way you can start doing your practicals. And there are two solutions to that. Either you find a big bag of money, so you can pay for everything, or you become Bob the Builder for molecular biology. And just to show, this is a poster that students made. So what they did, they built using a flower vase, they built a bioreactor. And with that bioreactor, they produced polymerase. And with that crude extract for polymerase, they could already amplify some DNA. So this was a pilot experiment to test the theory that home-built equipment would allow to produce these reagents. And this is what we want to scale up with the team project that we currently have and see how we can implement this in Kenya and in Ethiopia. And with that, I'm at the end of my talk. Thank you very much for your attention. If you still have questions or remarks, you can always send me an email. And I hope you enjoy your this VLIOS community talk. And I hope it's inspirational for you and that we see a lot of change on this planet happening. Thank you very much.
Hello everyone, I am Fanos Tadesse, a PhD student of Catholic University of Leuven. My promoter is Professor Jan Paeshuyse, and I'm working in his laboratory, host pathogen interaction. I'm working on foot and mouth disease vaccinology and epidemiology in Ethiopia. Livestock in Ethiopia are very important because they are contributing about 16% of the agricultural GDP, and there are societies which have a very good link with livestock products and the livestock services like milk and the draft power from that animal, and the services which we just get from animals are very important to serve the community, specifically women's. And the livestock products such as milk and meat are very important for children because there are a problem of malnourishment in the sub-Saharan countries. Foot and mouth disease is one of the most contagious disease that affects cloven-hoofed animals, both domestic and wild animals. And it's a very economically important disease, and it's caused by a virus called foot and mouth disease virus. And it's a very devastating disease, and it's prevalent in most parts of the world, specifically the sub-Saharan Africa, Asia, and the Middle East countries, and it is caused by 7 serotypes: serotype A, O, C, SAT1, SAT2, SAT3, and Asia 1. And of course, there are about more than 60 topotypes. And in the presence of these topotypes and serotypes, we have also a number of domestic and wild animals which are very susceptible. And the control of this disease is very complex because of the existence of 7 serotypes and more than 60 topotypes, which are not having a cross protection, which means infection or vaccination by one serotype cannot protect a second infection by another serotype. So this disease is a very dangerous disease. Economically, it is a devastating disease, and it prevents us, specifically for least developed countries, the least developed countries, it prevents export of meat and milk, even animals, to a developed world. So trade embargo is the most important thing that affects those least developed countries, in addition to the loss of animals from the draft power, and as well as reduction of milk production from these animals. So this is the most important and the most devastating disease ever known. And with respect to the challenges that we have in terms of diagnosis and research, I can say that the equipment, reagents, and laboratory personnel are very short. Of course, we need to work on that, and the KULeuven project aims to just strengthen that part, and we were doing on training laboratory personnel, students with regard to that, and we are also planning to produce some reagents locally.
My name is Samson Leta. I work for Addis Ababa University College of Veterinary Medicine and Agriculture. I'm an associate professor of spatial epidemiology and biostatistics. I'm here to talk a little bit on foot and mouth disease. Foot and mouth disease is one of economically important viral disease of cloven-hoofed animals with a significant economic loss. So we have outbreaks of foot and mouth disease every year, even if we are vaccinating our animals using a trivalent vaccine. The KULeuven FMD project have been doing good, especially in creating awareness, using training farmers and experts, and also on the surveillance, as well as also in fulfilling laboratory equipment, especially the molecular laboratory facility of College of Veterinary Medicine and Agriculture. Thank you.
So my name is Dr. Joseph Kamau, a senior research scientist here at the Institute of Primate Research, and also an adjunct lecturer at the University of Nairobi. My research interest crosses different areas. I specialize in One Health, pathogen discovery, and also in food security in terms of either antimicrobial resistance, biosafety and biosecurity, and diseases that affect mostly or touches on the food security. Livestock industries is a big thing in the country and the region, basically in Africa, and 10 to 12% of the national GDP or the economy is basically derived from the livestock production or livestock industry. That's in terms of either meat production, animal products, and also it comes to basically either in terms of the animal itself, usage, or the products that we get from the animals. As for the economy, 10%, which is quite a big chunk of the national income, comes from the livestock industry. And around 40% of the basically the Ministry of Agriculture, around 40 to 42% contribution in that ministry is purely from the livestock industry. So you can, in terms of contribution, when it comes to livestock in the drivers of the Kenyan economy and the region, livestock plays a big role. Livestock production in Kenya, like almost 70 to 80% is basically done by the pastoral communities. Given the ecological parameters in the country, most of the Kenya, it's only around 30%, one third of the country is arable land, where both the crop production and livestock production is kept. But almost 60 to 70% is arid and semi-arid areas. And in that areas is only livestock production that mostly are basically, are produced or kept there. And the majority of the community that basically produces these animals are mostly the pastoral community, which are basically vulnerable given they're basically from they live in those arid and semi-arid areas, quite harsh environment, remote areas where it's not accessible. And in terms of the economy, or basically the economy in that region is quite low. And so this community is very vulnerable. And at the same time, these are the livestock production again, or the animal that again providing almost 10% of the economy. So the economy being provided by the, again, vulnerable population. And in these communities, most are the women basically who keeps, because the opportunities for job employment or work is basically kind of gender specific, and most men tend to get employed. But in the pastoral community where the women are left home, or the children, so those are the people who basically keeps, or basically tends to, keep the livestock, or basically, rear those animals and make sure that they are okay. So whenever we have this again, issue of the disease burden, whether it's across the boundary or best within any outbreak that affects the animals and the livestock per se, basically these are the community or the population that tends to be hit hard. And so it's this need basically to see that any intervention that can basically bring up their stable income is taken care of. And there's none other than to make sure that the disease control or prevention is handled well. The disease knows no boundary, and transboundary diseases basically in our region, and given our culture, behavior, and practices, so there is no boundary for disease. So the disease that found across the border, either Ethiopia or other countries in the region. In Kenya and Africa at large, we have a myriad of challenges when it comes to research, not only just in diseases, or types of research that we undertake here. And this can be said in three ways. One, infrastructure challenge. When it comes to the equipment availability and accessibility, we have problem because even simple things like sequencing, you have to ship your samples across the border, either to the Europe or basically the US. To be able to do that simple sequencing. Then the other thing is basically the personnel. We have limitation when it comes to the training, or basically the specific or specialized training to handle specific areas in research. So you find that we don't have that basically enough, I could call emerging technology or training in the emerging technology that you could be able to use to handle some of the emerging challenges that we are facing in research. And the third one, and again, which I could call the most important part, is basically the supply of consumables and reagents. All the two, we have to import them from abroad, either Europe or basically Asia or US. They become very expensive. So something that costs maybe $30 in Europe may cost, by the time it gets here, maybe it's costing almost like $120. So it becomes so expensive to maintain or to carry out research. So it's not sustainable. And so these are some of the challenges that we are facing here. And we think KIT, FMD program, the project that we are currently undertaking, is trying to answer some of those challenges. One, we are getting our personnel in terms of the student and the technician going to be trained in Belgium, University of Leuven, basically get to know or transfer some of the technology back here when they come back. And the second one is we are hoping that before the end of the program, some of the things that we had started, like basically seeing how we can be able to produce some of the consumables, like especially the tag polymerase. That's very key and very expensive, that you can be able to transfer some of those technology here and start producing some of the consumables. That will be very key to sustain research when you go further or basically forward. And the third one is basically the additional kind of collaboration that we are doing with the North and specific KULeuven, where we can be able to share experiences in terms of basically training and other areas that we may be able to collaborate, that will be able to kind of help and strengthen our research here. And so we think the program is very timely, very helpful, and hope by the end of the project, some of those challenges will be either have been reduced or minimal, or kind of basically find a way to handle them in a better way.
Nashipai, Karinten. I'm a veterinary surgeon residing in Laikipia County, a place called Endana. I serve the pastoral communities. I practice in this pastoral setting. I mostly do disease surveillance and clinical diagnosis of diseases. Today we want to talk about foot and mouth disease, which is a major challenge in this pastoral setting because of the unrestricted movement of animals. Foot and mouth disease is caused by a foot and mouth disease virus, which affect all cloven hoofed animals. It is transmitted from wild animals to domestic animals via the feeds, the beddings, and contact. When the animal gets foot and mouth disease, they get lesions in the mouth and on the toes, so it limits feeding and movement of the animals. And eventually, if the viral load is high, the animals will die and they continue spreading to other animals. It's an economically important disease in this pastoral setting because it's majorly seasonal in these areas. It occurs mostly during the dry period, but lately it has been occurring throughout the season, so it's causing huge economic losses to the pastoral communities. Of late, it's been affecting both cattle, sheep, and goats, and it's also being seen in the wild animals. So it's a disease of concern right now. Foot and mouth disease is a transboundary disease, and in this pastoral setting, the disease has been in rise due to the movement of animals from Samburu, Marsabit, and Laikipia. So there's no control of the disease, and it's mainly due to the animal movement into our domestic animals are invading the wildlife sites, and we are, as it is known, buffaloes are the reservoirs of foot and mouth virus, so it's spreading to the domestic animals, and disease has major impact as it causes death of animals, it causes restriction movement, trades, and mostly it causes storm of abortions. This disease is perceived by the community to even affect human beings, as most of the time, if people consume milk or meat from animals infected by foot and mouth disease virus, the community perceives it to cause abortions in pregnant women. So according to our One Health, they are currently conducting massive vaccinations to try and mitigate the effects of the foot and mouth disease virus in human beings and also in people.
Hello, my name is Christopher Kariuki. I'm affiliated to the Host Pathogen Interaction Laboratory at the KULeuven. My interests are mainly molecular and structural biology, as well as the teaching and training of these techniques and the tools that they contain. My involvement with the KIT FMD began at the inception of the consortium, whereupon we met with Jan, Professor Paeshuyse, at a conference or a workshop that we had organized together with the Inter-University Program for Molecular Biology at the VUB, and this was in 2017 in Kenya. My role within the project has been mainly on the training of the different students who are involved in the project, as well as formulating the different topics and advising on the different aspects of molecular biology and structural biology that are required to be used in the project. One of my main interests is on the aspects of reagent production, particularly for the Horn of Africa region. This would ensure independence on the supply of some essential reagents such as Taq polymerase. And I'm very happy to have been involved in this project, especially in the inception of the bioreactor, which was used to express some Taq polymerase. And we are currently working on enhancing and improving this bioreactor, and also incorporating other polymerases that can be expressed with this bioreactor, that is open source polymerases. For this, we'll ensure our independence from expensive reagents.
Hello, my name is Eyerusalem Fetene. I am from Ethiopia and I am one of the master students attached to the Villares funded KIT joint project. I graduated my first degree in Doctor of Veterinary Medicine from Addis Ababa University College of Veterinary Medicine and Agriculture, and I pursue my master's degree in veterinary epidemiology. For my master's thesis, I have been doing this research entitled Detection, Identification, and Transmission Dynamics of Foot and Mouth Disease, which is one of the most devastating livestock diseases in the country. And this project has been helping me by providing necessary equipment, laboratory reagents and kits, materials for the sample collection, and also the project has been covering my field trip expenses. Thank you.
Okay. Yeah, if there are any questions, we'd love to address them. I do have a few questions. What is the situation at this moment in Kenya and Ethiopia regarding foot and mouth disease? Can you answer that? Is that maybe?
Fanos, can you update on Ethiopia?
Yes, currently, the disease is there. Of course, it happens year round from October to March, end of March, and we have 4 circulating serotypes currently: O, A, SAT1, and SAT2, and the dominant is serotype O, followed by A and SAT2. Sorry. So yeah, the disease is there, and all KULeuven domestic animals are susceptible in the current situation.
And then can it also what is the situation of the humans then? Because we are talking about animals, but I heard some cases that it can also go over to people as well. So what is the situation on that? Are you aware of that?
Well, the disease is not that much zoonotic, and it might have like signs, but it's not a very serious one. But there is the same disease in human, which is called hand and mouth disease. Yeah, but this one is a different one caused by another virus. But this one is not the zoonotic disease, and its importance is mainly economic importance.
Perhaps Joseph can also add a bit on that.
Yeah, so my name is Joseph Kamau from Kenya. So we have a different kind of experience here, as my student Nashipai did mention in her video. Initially, we had this seasonal kind of outbreak for FMD. Right now, it's kind of cutting across every season, even the dry and the wet season. You find the outbreak, those kind of different foci of the country. And because of the movement of the animal, especially during the dry areas, during dry time when they are trying to seek for pastures, so they kind of goes across the different corridors, both the national parks where they have the wild animals and other areas. So outbreaks is now kind of everywhere. And one of the things fun that when I'm discussing this with Chris, we did some work. My student did some work of sequencing. And some of this isolates that were antigenically, maybe detected as A, as serotype A. When you do the sequencing, there are basically different kind of serotypes. So maybe it becomes maybe SAT1. So meaning antigenic kind of is having issues when it comes to the identification of the virus. But as Nashipai, my student mentioned, which is, I think, Professor Ann and Chris, we may want to discuss this more. We were in the field and we found that the pastoral communities who are basically have been experiencing this disease for so long, traditionally or basically through the traditional knowledge, they have understood that when they have FMD outbreak, women who are expectant are not allowed either to take the animal products from that animal, either milk or the meat, because it causes abortion, or they let it through the abortion. So basically, you can imagine this is a community that they do not grow crops, so they rely most of their food is coming from the animal. And this is an expectant mother who require nutritional support. At this time when the animal, because it's having the FMD, that lady will not access the milk or the animal products from that animal. So even pushing that lady even to more malnutrition. And that's why we have maybe mortality in children's high, because if it's happening, maybe say in a certain area, maybe in one of the counties, it means then the women there who are expectant are not going to get access to the milk and meat, which is one of the biggest source of protein in that community. And it's one of their livelihood. So when I was discussing with them, we were trying to, I have seen a publication from, I think, West Africa, where also they have kind of an association of the same FMD being associated with abortion in women. So also this one we experienced when we went in the field with my student. We have currently no mechanism. We don't know what that's the mechanism behind that. It's something that we want maybe to discuss more and see whether we could be able to pinpoint exactly what's happening. and why it's maybe related to causing abortion. So it's something that is now becoming more prevalent than just in humans, in animals, given now that it's a threat to expectant women who are more vulnerable, and we are pushing even more, basically limiting their source of protein in per se, which is very important when expectant, when the lady is expectant. So I guess it's an interesting area that we should do more research, try to find what exactly is happening, get more data that supports exactly what might be happening, and maybe that's something that I would want to discuss with Professor Jan and Chris to see exactly how we could go about it. Thank you.
Thank you. Joseph, for sure we should discuss that.
Yeah.
Are there more questions?
For me, I would say the project has been very nice because of what Jan has talked about. I'm very interested in the independence of independence on its self-sufficiency for the reagents, and the knowledge that this was basically what was done in Western Europe in the early parts of the molecular biology revolution. For me, this is interesting because if we could replicate that kind of situation within Africa, or rather within our region of Africa, then we can achieve our self-sufficiency. Instead of just being consumers, we can build a body of knowledge and a body of expertise within the people who know how to resolve these reagent problems. And in the process of also resolving the reagent problems, you'll find that you also create a body of knowledge in which people now understand at a practical level what exactly they are doing, not just following protocols that they have just read in papers or heard about in books. So for me, participating in this kind of a project has been a very rewarding experience.
Thank you. More questions or thoughts or suggestions?
I do have actually another question. Why did you limit your research to the Horn of Africa? Why not broader? Doesn't exist there outside of the Horn of Africa, or
Sure.
or are there any new ideas of broadening it up?
That had to do with the limitations of the countries that were eligible for US support.
It's true, there are 20 countries that are partner countries that we are working with, 17 by now, by the way. And indeed, Kenya and Ethiopia are one of our most important countries in our whole portfolio. There's also Vietnam, of course. Is there any Vietnamese here in this room? Because it would be interesting to hear the situation in Asia as well, maybe broader up in Asia. I mean, we are not that many in this room, so please raise your voice if you have something to say. It's easier. I see also Professor van Orshoven who's in the room here. Professor van Orshoven, you're also involved in Burundi, isn't it? So do you have any idea what the situation is there in Burundi regarding foot and mouth disease? Professor van Orshoven, I see you're there. Maybe he doesn't know. maybe anyone else knows what the situation is in outside Africa then, because actually that was my initial idea, of course. The limitation of the partner countries. Burundi, by the way, is also a partner country together with Rwanda and Congo. So there's still plenty of possibilities. Tanzania as well. So
I think the situation there is probably fairly resembling Kenya, the situation in Kenya and in Ethiopia. So I guess that we can expand to those countries.
Okay. Well, in the case if there are no questions, then I think we have come to an end of this. Are there any other remarks, or we still have, 10 minutes? It would be new that we have a very short, but why not? Because usually it's always longer than 1 hour. But it can also be shorter, of course. So if there are no comments, if there are no specific questions.
Christoph, I see a question in the chat. There's someone in Jedala Robe Godana from Ethiopia, Bulehora University in Ethiopia. So he's asking, how can everybody intervene for a solution, since the life of the community, the pastoralist community, is based directly on livestock, and the livestock is in high risk of this disease. So what is the solution? So I think that is probably a question that Fanos can touch on, probably.
Yeah. Well, I know that there is a problem in the Boran Pluto currently. There is a drought, actually. So I think Jadela is referring to that specific drought problem, if I'm sure, with regard to disease control, because animals, they don't have grazing and water by this time. So I don't know what to say, but yeah, it's very important to tackle with, especially in this situation, to control the disease or to just cure the animals or something like that. But this project is a very small one, and it's impossible to say something on his question. So I think, yeah.
And do you?
Yeah.
Good morning, everyone.
Okay.
I don't know if you can get me.
Yeah, we can hear you, Joseph.
Okay. I'm a Bandit Joseph, PhD student at the Post-Pathogen Interaction Laboratory, also working in African Fever. And before joining the lab, I also had the opportunity to work with cattle in Cameroon. And foot and mouth disease is also a very big problem over there, which we also have seasonal outbreaks. But so far, research in my country is really limited in that domain. So I think it would be also an opportunity for me to ask if Cameroon, on the other hand, can also be part of this support, the South Project support, so that such initiatives can also be undertaken in Cameroon. Thank you.
This was a very bad connection. I'm sorry, I didn't
hear a lot.
You didn't hear neither. Okay.
I think that Joseph was complaining that Cameroon was not part from the country selection. They're eligible for PhD grants, but they're not eligible for project support to US. And that,
that's something you people are supposed to check on our website. So on our website there is all the information. It is not us who are deciding it. I mean, as I explained at the beginning, there is this cooperation of 5 universities and 13 university colleges, and together it is decided also together, of course, with the ministry, because we are fully financed by the Ministry of Development Cooperation, and it's together with them that it is decided. There is also our French sister organization, Addis. And if I'm not mistaken, I think they are working on Cameroon. So if you have, please check their website. So there might be possibilities to work with Cameroon. But it's true, it's very limited, but we also have very limited money. And that's why I'm happy that we can invest this little money in this kind of research. Okay, any other questions or remarks?
My biggest question is maybe to Jan, to Fanos, and to Joseph. Do they foresee a situation when foot and mouth will be controlled within our region? As in, this is more of a philosophical, or rather, a question that I don't think
that is not going to be resolved right now, but what would be the major steps that they think would lead to this? So maybe they can give their views.
I think first step in such a complex disease situation, so disease ecology, is that getting your vaccines matched to the circulating strains and trying to implement that as a first line of prevention together with biosecurity, and to implement biosecurity measures, that is a feasibility. And then from that point onward, it becomes more complex, because if you look at pastoral communities, biosecurity and pastoral communities quite difficult to combine. Animals are mobile, they're migrating. And if you then look at the disease ecology with species like the buffalo that can be carriers or reservoirs, or at least transmit the disease from the wildlife back to the domestic cattle, there are things that you can decide, but it's difficult, I think, to kind of either start modifying cattle raising practices like pastoralism in order to fight or eradicate foot-and-mouth disease. So I think there we should look at another or a different strategy. I think that Fanos can also comment on that with the difference in susceptibility. If you saw in the presentations, many of the animals that were depicted were to Ethiopia and Kenya exotic breeds. And if you look at, or if Fanos has made some field observations where he established the difference between susceptibility, or at least between the pathology that's observed in different breeds. Can you comment on that, Fanos?
On my observation, I saw that those exotic breeds have a very severe pathology of foot-and-mouth disease, but the local breeds are not that much showing the pathology. They have the disease, but the pathology is not severe. So from that, if I'm not optimist, I can say breeding policies should be checked again. We, of course, that will be the last option. Just vaccination and biosecurity measures should be first applied in this situation. And as Professor said, the vaccine that we have currently should be matched with the circulating strain. Otherwise, it's like injecting the water or something like that. If the circulating strain, if the vaccine does not have a match with the circulating strain, I think we cannot see protection, as we said. And of course, it's complicated by the presence of domestic wild animals in the area. So that should also be considered. And I think a number of factors should be considered to control this disease in that area.
I think also there for the pastoralists, by looking at which breed they have and how it co-evolved with foot-and-mouth disease pressure, that's important also as a strategy to reduce or to minimize the impact of foot-and-mouth disease.
Okay, it's 11. So the hour is past. I would like to thank warm-heartedly my colleagues, Chris, Fanos, Professor Paeshuyse,